Sorry. Good evening. Good evening. Okay, now let's continue. So if every one of you very quickly recall in the yesterday class, we have started discussing about the process of the eukaryotic transcription. Correct, na? I want every one of you quickly answer my question. If you recall, there are two type of promoters are present. Correct, na? One is called proximal promoters. Another one is called as what we can call it as core promoters. Is it? Now, my question to all the students is, among these two, which of the promoters are present both on the upstream direction and on the downstream direction? Every one of you, good evening. Please continue. My question is, which promoters are located both on the upstream also, both on the downstream also? What are those? For every one of you, of course, which is the proximal promoters. Please don't get confused with that particular point. And friends, when you look into this particular core promoters, what we discussed, friends, they are present only on the left-hand direction. Okay, that's perfect. Okay, now my simple question to all of you, tell me in one single line. We have studied that there are two types of proteins are there. One is called enhancers, another one is called as silencers. I have very clearly told to all of you friends, enhancers enhance the process of transcription, silencers suppress the process of the transcription. Now my question to all the students is very clear. How does the silencers can suppress the transcription? Or how does the silencers can silence the process of the transcription? Just give me a single line answer. How does the silencers can silence the process of the transcription? Every one of you quickly. Undoubtedly, friends, the silencers are very, very good. The silencers are going to silence the process of transcription by converting the DNA into a condensed form, by converting the DNA into a coiled form. That is what exactly, friends, we by converting into a heterochromatin form. So that is how they block the process of transcription. In the yesterday class, we have started studying about this particular slide and we stopped it. You remember there are transcription factors which are involved in the process of the transcription. What I told to all of you in the yesterday class, friends, whether it is a process of initiation, the process of elongation, and the process of termination, all the processes are exclusively depends on the transcription factors only. If everyone quickly recall my words, I told to all of you very clearly that there are two type of a transcription factors. One is called general transcription factor and another one is called, friends, promoter specific transcription factors. If you quickly recall my words, general transcription factors, what did I told you? Of course, they are useful, but what I told you, they are involved in the basal level of the transcription. Right. Promoter specific transcription factors are the one which are specific for every single gene and they can show the maximum level of the transcription. Please don't get confused. So listen to me 20 minutes continuously without having a confusion and try to understand what exactly I'm trying to explain to all of you. There are different type of general transcription factors are there. Let's start. We have started now the process of initiation. Good evening. Please continue. We have started the process of the initiation. So friends, in the initiation, what are the type of the transcription factors that are involved? Every student remembered. So see this, I am calling it as a TF2. TF represents transcription factor. Then why I am using the word Roman letter 2? 2 represents because all these transcription factors are helpful to the RNA polymerase 2. That's why we are calling it as the transcription factor 2. So there are many transcription factors are there. First one is called transcription factor 2D and the second one is called transcription factor 2A and next one is called transcription factor B and the next one is called as transcription factor E and the next one is called transcription factor 2F and the next one is called transcription factor 2H. So friends, these three transcription factors that plays a very crucial role in the in the process of eukaryotic transcription. I'll explain to all the students one by one very briefly and elaboratively. Just try to understand what exactly going to happen here. Focus, everyone. Just focus. Just focus. So I will explain step by step. Just listen to me. First one, transcription factor 2D. Remember my statements as it is. We have started the process of initiation already, friends. We have started the process of initiation already. Transcription factor 2D. It is exclusively made up of two proteins. One is called TBP protein. Another one is called TAF protein. What is the full form of the TBP protein? Tata binding protein. And what is called TAF protein? Transcription activated factor. Once again, remember, TBP represents Tata binding protein. TAF represents transcription activated factor. Here, every one of you listen my statements very correctly. The TBP protein will come and associated with what? It will come and associated with what we can say that Tata box. That's why we are calling it as the TBP, Tata box binding protein. And where this particular TAF attaches to the INR region. Every one of you know very well, friends, the process of the transcription exclusively start from an area or a region. We call it as INR. Exactly from that particular to the INR, TAF will go and attach. Such a simple step it is. So once again, I'm telling you, transcription factor 2D contains TBP, TAF. TBP attaches to the Tata box, TAF attaches to the INR. You got the point. Then the next one, transcription factor 2A. I can tell you in a simple words to all of you, transcription factor 2A is extremely important to maintain the stable interaction. Remember everyone, to maintain the stable interaction between the transcription factor 2B and TBP. Remember once again without having confusion. What's the function of a transcription factor 2A? It is responsible for maintaining the stability, maintaining the stable interaction between the transcription factor 2B and TBP protein. Don't get confused with that. Now listen. So what is the function of, first I will explain all, then I will show you the diagram. Anyway, let's quickly look into this. Look into this, every one of you. What I told you, friends, transcription factor 2D contains TBP protein, contain TAF protein. What did I told you, friends? TBP protein attaches to the Tata box and TAF protein attaches to the INR. And we studied there is another factor called as transcription factor 2A, which is exclusively required to maintain the stable interaction between the transcription factor 2B and TBP. We got the point correctly. Now all the students listen to me. Now the third one is called transcription factor 2B. Every student remember my statements without having confusion. Just remember, transcription factor 2B is required for start site selection. In a simple words, where exactly the INR is present, where exactly the initiation region is present, that can be recognized by this particular transcription factor 2B. Please don't get confused with that. Then friends, there is another very important factor. I can say to all of you, this is the most important factor which is associated with the RNA polymerase enzyme from the initiation till the termination. In every step of the transcription factor 2F is present. Listen, every one of you correctly. Listen, transcription factor 2F originally identified as RNA polymerase 2 associated protein. I can tell you in a simple words to all of you. You remember, let's consider a prokaryotes. This is a RNA polymerase enzyme and this is a promoter. And what is this? This is the sigma factor. Sigma factor will help the RNA polymerase enzyme to guide where exactly the promoter is present. Simple point it is. Similarly, friends, remember my words, transcription factor 2F, it is also exactly having the activity like a sigma. So that also they can rise in the form of a question. So transcription factor 2F, which is having a sigma like activity, friends, It will help the RNA polymerase enzyme exactly bind to the promoter. Very specific binding to the promoter can be mediated by the transcription factor 2F. Then the next important factor, transcription factor 2E. I will tell you in a simple words to all of you. Listen to me. Transcription factor 2E is the factor which is required for the recruitment of H. Remember what I'm trying to explain you. Transcription factor 2E is majorly required for the process of the for the recruitment of transcription factor 2H. Every student remember my words now. Now, friends, the transcription factor 2H having three functions. Listen, all the students. It is having helicase activity. It is having kinase activity. It is having ATPase activity. Name itself indicating, right? Tell me, helicase activity means what, friends? It is the one which is involved in the opening of the double-stranded DNA. You know it very well. And what is the kinase function? Kinase is the one which is involved in the adding the phosphate group. To where? To the RNA polymerase enzyme. And ATPase, which will donate the phosphate group. Now, before this, I want all the students has to understand one important statement here. Listen to me very carefully, friends. This is a very unique point. Imagine this is a RNA polymerase enzyme. This is called, it is a protein, right? So protein having N-terminal domain and protein having C-terminal domain. All the students answer my question. I hope you all remember. The C-terminal domain of the RNA polymerase 2 enzyme having how many amino acid sequences? Any student remember? How many number of amino acid sequences are present on the C-terminal domain of the RNA polymerase 2? Jyoti, Varalakshmi, forgot everyone. How come you forgot? Srinivas, Venu. There are 7 amino acids are there, right? Which we generally called as a heptad sequence. Very, very good. Good, Shubham. There is a presence of 7 amino acid. So friends, all these 7 amino acid undergo the process of phosphorylation and dephosphorylation. Now I am going to tell you an important statement that all the students remember my words very correctly. Whenever during the process of initiation, I am using the word initiation. During the process of initiation, these all amino acid present at the C-terminal domain undergo minimal phosphorylation. What is the statement I'm using? Minimal phosphorylation. But friends, when it comes to the process of elongation, they will undergo maximum phosphorylation. When it comes to the termination, they will undergo the process of dephosphorylation. So you have to understand, when the amino acid present in the C-terminal domain added by the phosphate group, which means that initiation is started, minimal phosphorylation. If all the amino acid undergo the phosphorylation, which indicates that maximum phosphorylation. Please don't get confused with that particular point. And termination, it will undergo the process of dephosphorylation. Don't get confused with that. Now you listen, look into the point correctly. Now you automatically understand the picture. Every one of you pay attention, try to understand the concept. Let's focus on one by one factor elaboratively. So the first factor, now the initiation is started. Transcription factor 2D contain TBP, TAF. You only look into this. TBP attached to the Tata box. TAF attaches to the INR. You got that point. Later, who will come into the picture? Transcription factor 2A will come into the picture. Later, who will come into the picture? Transcription factor 2B. I hope you all remember, right? Transcription factor 2B is required for the start site selection. It is required for which process, friends? It is required for the start site selection. And now all the students try to understand my statements very correctly without having a confusion. Now, who is coming into the picture? RNA polymerase. Now, I'm sorry. Now, who is bringing this RNA polymerase enzyme? Transcription factor 2F. the transcription factor 2F always associated with the RNA polymerase. Every time you see this is the RNA polymerase, 2F will be associated all the time. Now, what the 2F is doing, listen to me carefully. 2F is guiding the RNA polymerase. I'm sorry. The 2F is guiding the RNA polymerase to recognize the promoter. Now we got that. Now who is coming into the picture? Transcription factor 2E. If everyone quickly recall, I told you transcription factor 2E is exclusively required for the recruitment of transcription factor 2H, which is a very, very important one. Now, if you look into this picture, you will get a very good idea. You can see this, right? The red color one. This is called RNA polymerase enzyme. Now you can see the picture. This is called C-terminal domain. Now you can see the picture very clearly that during the time of initiation, you see this. There is a minimal phosphorylation is happening. What is the meaning of a minimal phosphorylation? Means all the amino acids are not completely phosphorylated. That is called minimal phosphorylation. Now, you all people has to remember a very important step is about the transcription factor 2H. What we discussed, friends, it is having helicase activity, it is having kinase activity, it is having ATPase activity. Now, you only look into the picture correctly. So, with the help of helicase activity, what it is doing, tell me, it is opening the double stranded. You can see this. See this? It opened the double stranded DNA because you know very well we require only one strand of the DNA as the template. So friends, it is compulsory we have to open the double stranded. Who is opening? 2H is opening. Okay. Now the initiation is started. But what I told to you, if the process of the initiation wants to be continued, what has to happen? The minimal phosphorylation occur. So who will add the phosphate groups? Of course, transcription factor 2H only, because it is having a kinase activity. And the question is very clear. Who will donate this phosphate group? That also done by the 2H only, because it is having the ATPase activity. Now you can see the picture very clearly, friends. Listen every one of you. When the helicase such as transcription factor 2H open the double-stranded DNA, did the minimal phosphorylation of C-terminal domain of the RNA polymerase enzyme, which indicates that the process of initiation is started. The process of initiation is started whenever the minimal phosphorylation occurs. Now, there is a problem. Now, I want you to compulsory remember what are the factors that are involved in the transcription factor, in the process of initiation. You no need to read this. I already told you, you can easily correlate them. Now, all the students listen. What are the factors that are involved in the process of elongation? This is more important among all. Now, all the students try to understand my statements very correctly without having confusion. This is the DNA. This is the INR. This is the INR. This is the RNA polymerase enzyme. Okay. Now, RNA polymerase enzyme starts synthesizing the RNA. There is no problem at all. Now, the point is that, listen all the students, whenever RNA polymerase enzyme is moving continuously on the DNA, you know, listen everyone, this is INR, right? From the INR, whenever the RNA polymerase enzyme travels 20 to 50 base pairs, RNA polymerase enzyme will stop immediately. Now, the question is that, friends, why it will stop immediately? The reason is that there is a certain passing sites are present. What are present? Passing sites. It's like a toll gate, friends. See, even though you're driving a car with 100 km/h, the moment you reach the toll gate, you have to stop immediately. Because your car can't move by crossing the toll gate without paying the fees, without paying the toll fees. Same thing happening here. All the students has to understand me. Whenever RNA polymerase enzyme started the synthesis of mRNA, It crossed the INR, reached 20 to 50 base pairs, and the moment it reached the passing site, what's happening now? Now, finally, RNA polymerase is here. It stopped immediately. Now, friends, tell me, if it stopped immediately, how does the process of elongation occur? Now, you remember. Now, whenever your car stopped at the toll gate, how your car will move? When the gate opens, car will move. How the gate opens? When you pay the toll fees. Simple. Same here also. How does the RNA polymerase enzyme will cross the passing site? The question is, friends, how does the RNA polymerase enzyme cross the passing site? Friends, I told you, right? It is having a C-terminal domain. You all the students look into the picture here. In the process of initiation, what happened, friends? In the process of initiation, minimal phosphorylation occurred. Now you can see the picture. At the passing site, the RNA polymerase enzyme undergoing the maximum phosphorylation. Whenever it undergo the maximum phosphorylation, friends, it can easily cross that particular passing site and continuously start synthesizing the mRNA. Now the question is here, how it undergo the complete phosphorylation. Now you all people listen to me correctly. I want you to understand my statements correctly. So right now we have studied in the initiation, there are 6 to 7 factors are involved. Similarly, friends, in the process of elongation also there are some factors are involved. What are those? Listen everyone. I always encourage all of you friends to remember the short names. Always remember the short names. It is called DSIF and another one is called NELF and another one very important one, PTEFB, positive transcription elongation factor B. You all people has to understand me very correctly. See, try to understand what I'm saying. Suppose your car is coming very fast. Example, 100 kilometer per hour. It is coming. The moment it reach the toll gate, the 100 kilometer car speed will become what? 0 kilometer. Correct, na? Then car also it is under the kind of a tension condition. So immediately what we will do? We will turn off the engine. Correct, na? We will turn off the engine immediately to relax it. Same thing happening here also. This is the INR, this is the passing site. Whenever RNA polymerase enzyme reaches the passing site, see, it is actually moving very fast, right? It is moving very fast, but whenever it reaches the passing site, it stopped all of a sudden. It stopped all of a sudden. Then it is also having lot of tension, lot of stress. So it has to stable. It has to become stable. Now the question is very clear, friends. How it will become stable? That's the question. How it will become stable, friends? With the help of a two type of factors. One is called DSIF factor, NELF factor. I always encourage you to remember the short forms. The DSIF and NELF, what they are doing, friends? They are just involved. Listen, everyone. Stabilize the RNA polymerase. Now, DSIF, NELF, they will associated with the RNA polymerase enzyme. They help the RNA polymerase enzyme to become stable, to not to move at that particular moment. But it can't wait at the passing site for a very long period of time. It has to move compulsory. Then you know what happened? This DSIF will activate another elongation factor, which is called positive transcription elongation factor. Now, you know what the positive transcription elongation factor is doing? It is recruiting another protein called as CDK9. So, friends, now that CDK9 will come and do the maximum phosphorylation. You can see this. You can see in the picture very clearly, friends. The positive transcription elongation factor B recruit the CDK9. Then the CDK9 is doing the maximum phosphorylation of the C-terminal domain. Then, friends, RNA polymerase enzyme can easily cross that passing site and it's continuously synthesizing the mRNA. So now the most important point, all of you listen to me correctly. There is a great possibility that we can expect the question from here also. Now, friends, you all know, right? This is the RNA polymerase. You can see, right? This is the mRNA. mRNA is coming out in which direction? 5 prime to 3 prime. So this is the direction where the nucleotides will be added, because 3 prime having a hydroxyl group. Now the question is very, very clear. How do we know? How do we know the RNA polymerase enzyme is synthesizing the correct nucleotides or not? How do we know? How can we conclude that? So friends, we can easily conclude with the help of a transcription factor called TF2S. They can ask a direct question in the exam. Which of the following transcription factor is involved in the proofreading or checking the errors, which is called transcription factor 2S? And remember, friends, they can also ask the question in the exam. Transcription factor 2S, it is also involved in the DNA repair mechanisms also. Just remember this point, I'll tell you in the DNA repair class. It is also involved in the DNA repair mechanisms. Now all the students listen to me very correctly without having confusion. So this is the direction mRNA is coming out, and proofreading is occurring from which direction? Backside. This is called backtrack mechanism. In this direction, we are checking whatever the nucleotides added by the RNA polymerase enzyme, is it a healthy nucleotide or not? If we identify that whatever the nucleotide that it is added, it is the wrong nucleotide. So what's gonna do? So what it is exactly doing now? Friends, it is immediately, all the students try to understand, it immediately stop it. Stop the transcription elongation. Now you can see, right? The transcription factor 2S will come into the picture. It will remove the wrong nucleotide. The moment it remove the wrong nucleotide, it is going to add the healthy nucleotides. Once it replaces with the healthy nucleotides, friends, immediately what is going to happen? You can see that mRNA can be synthesized continuously. Now the question is very, very clear. Every one of you try to understand my statements very correctly. When it can stop. So now you listen, try to understand. The mRNA is synthesizing continuously. RNA polymerase is also moving continuously on the DNA molecule. Now, when the RNA polymerase enzyme will stop, again, the point is very easy. Whenever the RNA polymerase enzyme reaches a termination site, where this particular termination site is present, friends, undoubtedly the termination site is present on the DNA. Now the question is here, how does RNA polymerase enzyme get dissociated? How does RNA comes out? How is this possible? It is possible just by a simplified mechanism called as dephosphorylation. I have told you, right? This is RNA polymerase enzyme. During the time of initiation, minimum phosphorylation, elongation, just now you have seen maximum phosphorylation. During the process of termination, entire C terminal, see, this is the C terminal domain of RNA polymerase. These all phosphate groups will be removed out. Once they removed out, RNA polymerase enzyme will become completely inactive. It will get dissociated easily from the DNA molecule. Please don't get confused with that particular point. Okay, every one of you try to understand once again what I'm trying to explain you here. Listen, look at this. This is the overall process. Elongation, termination, and release. It is a very, very easy process. Now listen. Once the RNA transcript is completed, means the synthesis of RNA is completed, the process of transcription has to be stopped. How does the process of transcription will stop, friends? Remember everyone, by the mechanism called as dephosphorylation. That's a simple mechanism. Now look into this. What's happening now? I'm summarizing the entire steps now. Look into this. First, which enzyme comes into the picture? Every one of you focus. Transcription factor 2D. It contains TBP protein, attaches to the Tata box. Transcription activated factor will go and attaches to the INR. Later 2A will come. Later 2B will come. Later 2F will come. What I told to all of you, transcription factor 2F is the true RNA polymerase enzyme. Correct, na? Because it is always associated with this particular RNA polymerase enzyme. Later, transcription factor 2E will come. then later H will come. You already understand. When the H comes, what happens? Minimal phosphorylation occur. In the process of elongation, what happens? You remember, right? Positive transcription elongation factor B is going to recruit the protein called as a CDK9, and it is doing the maximum phosphorylation. It is doing what? Maximum phosphorylation. Whenever it does the maximum phosphorylation, every one of you listen my statements. So now it reached the termination site. Now what has to happen? Now I have to remove all these phosphate groups. Once we remove all these phosphate groups, RNA polymerase enzyme will become completely inactive. It fall down. It will dissociate from the DNA and mRNA come out very easily. So in a simple words, the termination is a very easy termination. Polymerase 2 is getting released by the mechanism of dephosphorylation. Such a simple step and easy to remember actually, right? Okay. This is what? The termination of eukaryotic transcription. Is it clear, everyone? Just say yes or no. If you have any doubt, please ask me. Is it clear, everyone? See, eukaryotic transcription is always important, friends, when compared to the prokaryotic transcription. After the class, I want all of you has to look into the slides at least once. By that, you never ever get confused with that. Right. So now what we are going to study, the most important where there is a great chances are there that you can expect the question, which we generally called as what, friends? RNA processing. Why does this RNA processing is very important for the exam? Because this is a twisted concept. There is a possibility that they can raise the question in order to make us confuse in the exam. Now you listen. Now, I strongly believe you all people remember, if you look into the prokaryotic organisms such as the E. coli, what we studied? Replication, transcription, translation, everything happens in the cytoplasm. You all remember that. But if you look into the, what we can say that, eukaryotes is a very complex organism. What's happening? In case of DNA replication happens where? Within the nucleus. Now where the transcription happens? Within the nucleus. So this is the mRNA. Now what is the point? The point is that whatever the mRNA that is synthesized within the nucleus, friends, that mRNA is not functionally active. That mRNA is not biologically active. That mRNA is a immature mRNA. Now, friends, how does the immature mRNA can enter into the cytoplasm for the synthesis of protein? Immature mRNA cannot synthesize the protein. Now, the question is very, very clear. How can we make this mRNA into a mature RNA? How can we make this mRNA to become biologically active or functionally active? So to make this mRNA to become functionally active, we are doing some processes. We are doing some modifications. And friends, when we are doing that modifications, we are doing that modifications after the transcription. Yes or no? After the transcription. That's the reason only, friends. We called it as a post-transcriptional processes. What do we call it as? Post-transcriptional processes. Never ever get confused with that. Now, listen, because it is after the transcription. Now, that is not the question. What is the question? Where all the post-transcriptional processes are happening. That's the question. Within the nucleus only. Most of the students will get confused here. Once the mRNA get completely matured, once the mRNA become completely active, then only it going to enter into the cytoplasm. Otherwise, it never ever enter into the cytoplasm. Always remember that particular point without having any kind of a confusion. Look into this beautiful diagram. All of you focus on the diagram. This is the DNA. Every one of you know very well, you have studied in the intermediate also. DNA molecule having what, friends? Exons, as well as DNA molecule also having the introns. You know it very well. Now the question is that exons are what, friends? Coding regions which code for a protein. Introns are non-coding, but all the students has to look into an interesting point here. When compared to the coding regions, non-coding regions are very large. You can see that the regions which can code for the proteins, they're extremely small, very, very small they are actually. Now look into the concept correctly. Now what's happening here? Now I want to cut this intron. I want to join this exon. That is okay, fine. Before that, listen everyone, before that, we have to add a cap and we have to add a tail. I will explain you briefly why we have to add the cap, why we have to add the tail. But okay, Sanvika, please disable your writing curve. Okay, now listen, exon and intron. Remember everyone, on the DNA only, friends, listen everyone, we are having a poly A site. Every student remember, poly A site. What is the meaning of poly? Poly means many. A represents what? Adenine. We are adding many number of adenines. How? By cut this side. That's an important point. We cut this side and then only we will add. And don't confuse that we are not adding the polyadenines to the DNA molecule, because from the DNA, RNA comes out. So what I mean to say, whatever the information present in the DNA, the same information coming to the mRNA. Yes or no? Of course, yes. So DNA contain exon, DNA contain intron, DNA contain poly A site. All these are all coming to the mRNA also. Now you can see this picture. mRNA also contain exon, mRNA also contain intron, mRNA also contain a poly A site. Now all the students look into this. Now what I'm gonna do first? First, at the 5' direction of the mRNA, I'll tell you in the next slide why we have to add this. At the 5' direction of the mRNA, we are going to add a particular cap. Correct, na? So what do we call it as, friends? Methyl 7 guanine. They can ask a simple question in the exam. Which of the following nucleotide is added in the cap? Which is the guanine? Which of the nucleotide added in the tail, which is the adenine? Please don't get confused with that point. Let me explain you. Now, why did I call it as the methyl 7 guanine? Because at the 7th position of the guanine, it is added by the methyl group. That's why I called methyl 7 guanine. Now remember, if I want to if I want to add the polyatyl, what should I do now? You remember, this is what we can call it as. We call it as a poly A site, polyadenylation site. So friends, what we are doing now, we cut this particular site. The moment we cut this site, then only we are going to add the number of adenines. So see this, we will add nearly 250 adenines. That's why we call it as polyadenylation. Please don't get confused with that particular point. So first I added the cap, later I cut the poly A site, I added the tail. Now the most unique step was splicing. What should I do now? I don't require any introns. So we cut the introns and we add the exons. So removal of intron and addition of the exons, what we call it as, friends, we generally call it as splicing. Never ever get confused with that particular point. Always remember that particular statement. Don't confuse here. Okay, now that we understand it very clearly without confusion. Now, friends, look into this beautiful picture. You will understand one thing very nicely here. This is RNA polymerase. You can see here. This is what, friends? C-terminal domain. You can see, right, which got phosphorylated. Now you can see this, the green color one which is coming out. What was that? That is only mRNA. So to the mRNA, first who will go and attach as a capping machinery? Capping machinery will go and add the cap. After adding the cap, every one of you listen my words. Look into this. Splicing machinery will come and they will try to cut the introns. After the splicing machinery, you can see that polyadenylation machinery will come and they will add that particular polyatyl. So capping, polyadenylation and splicing, three together we can call the post-transcriptional process, or we also call, friends, this is most important, we also called as a pre-mRNA processing. Sometimes In the JL kind of exams, they will also say conversion of hnRNA to the mature mRNA. This is also called as pre-mRNA splicing. This is also called as post-transcriptional process. Why did I call it hnRNA? Because it is having a lot of introns. That's why I can call hnRNA. I can convert it into mRNA. That is what we can call the mRNA processing. Now let's look one by one very elaboratively. So first what we have to do, among all the capping is little confused and splicing as well. So I want you to understand the process very correctly without having a confusion. First one is called cap. Remember my words very correctly, friends. First one, what do we can call capping? Listen to me very carefully without having a confusion. First question, this is mRNA. This is the 5' direction. This is the 3' direction. Exactly to the 5' direction, I am adding the methyl 7 guanine. All the students listen. This is the guanine, right? So to the mRNA, I am adding this guanine. Why did I say methyl 7 guanine? You can see that the guanine exactly at the 7th position, it is added by the methyl group. That's why we can call it methyl 7 guanine. Now the question is that why we have to add the cap? That's the question. What is the function of the cap? They can give the question in the exam by saying that all of the following are the functions performed by the capping except. So you all people has to remember what are the major functions of the capping, why we have to do it. This is a very important, friends. They very good. So the capping protect the mRNA from where? They protect the mRNA from the process of degradation. Never ever get confused with that particular point. And the next point is that they enhance the translatability of mRNA. That's a very important statement, friends. You will understand in the process of eukaryotic translation. All the students just keep this point in your mind. 5' capping and 3' polyadenylation. Remember my words. Both are exclusively required. Both are exclusively required for the translation in eukaryotes. How they are involved in the translation, we will study directly in the translation, but remember that point. So 5' capping protect the mRNA from the degradation by lot of vulnerable enzyme, degradative enzymes. Now very important, friends, just imagine 5' capping is over, 3' polyadenylation is over, splicing is also over. Now the question is that the mRNA has to move from the nucleus into the cytoplasm. How does it will move from nucleus into the cytoplasm? Through the nuclear pore. Everyone know it, nucleus having a pore-like structure. Now the question is very clear. How does it will move? That's the question. Again, by the capping only. Capping is the one which is enhancing the transport from the nucleus into the cytoplasm. That also an important point that you have to remember. Another point, they enhance the efficiency of splicing. What is the meaning of efficiency of splicing? You can see this picture, no? The moment capping is over, who is coming? Splicing machinery is coming immediately. Now the question is that who will recruit the splicing machinery? Capping only. So capping machinery will do the capping. After the capping is over, it is going to recruit the splicing machinery. Please don't get confused with that particular point. Now all of you listen my statements very correctly. This is the most confused concept. If you understand this, automatically you will get the concept. Okay. Now every one of you focus. Just listen to me correctly. This is mRNA. mRNA having nucleotide, first nucleotide, second nucleotide, third, fourth, fifth, so on. So friends, to which nucleotide guanine will come and attach us? So mRNA, it is in the 5' to 3' direction. You know it. This is the first nucleotide. Tell me, first nucleotide contain what? First nucleotide contains sugar, it contain a base, and it contain a 3 phosphate groups. You got the point. This is the first nucleotide. So exactly to the first nucleotide, we are adding the guanine. Correct, na? Now, every student remember, if every one of you look into the picture correctly, this is the structure of a guanine. Tell me, how does the guanine look like? Guanine also look like same. It contains sugar, it contains base, and it contains the phosphate groups. There is no doubt about that particular point. Now all the students try to understand what exactly I'm saying. Guanine also, at the 5th carbon, how many phosphates are there? 3 phosphates are there. Now the first nucleotide of the mRNA, at the 5th carbon, how many phosphates are there? 3 phosphates are there. So here also 5th carbon contain 3 phosphates. Here also 5th carbon containing 3 phosphates. But the point is that we can't add all the 6 phosphates. Out of 6 phosphates, we remove 3 phosphates and we will add 3 phosphates. Now you see this, what I have written. You can see the picture very clearly. You can understand it very clearly. This is what? Nucleotide 1. This is the nucleotide 2. You can see that. And see here, this is the guanine. So guanine having 3 phosphate, first nucleotide also having 3 phosphate. But total, how many we added? We have added only 3 phosphates. And how many we removed? We removed 3. We will discuss how we will add 3, how we will remove 3. Now, so see, that's what I have written. Cap means addition of the methyl 7 guanine via 5' triphosphate bridge. And friends, why did I use the word 5' to 5' triphosphate bridge? What is the reason? Why did I use that word? Because at the 5th carbon, 3 phosphates are there in the guanine. At the mRNA first nucleotide, 5th carbon also having 3 phosphates. But out of 6 phosphates, I have added only 3 phosphates. How the 3 phosphates connected? Via 5' connection. That's why we can call it 5' triphosphate bridge. No need to confuse. It's very easy. Now, same thing I'm explaining. See this? This is the first nucleotide of mRNA. This is the second nucleotide of mRNA, third nucleotide, and so on. What is this, friends? This is the guanine. So this is the 5th carbon of the guanine. And this is the 5th carbon of the first nucleotide. How many phosphates are there? 3 phosphates are there. What happens to other 3 phosphates? We will discuss that. And all the students, listen, friends. We just now studied, right? Friends, guanine at the 7th position, it is added by the methyl group. That's why we can call it methyl 7 guanine. All the students has to understand me correctly. Since guanine got methylated at the first nucleotide, you know what do we call it as? Cap 0. Friends, don't get confused. This is the mRNA. This is the 5', 3'. Here only I have added the guanine, right? To the first nucleotide, I added the guanine. And at the 7th position, it is added by the methyl group. This is called cap 0. Now, this is the first nucleotide of mRNA. Every student remember, first nucleotide of the mRNA also added by the methyl group at the OH group. You know, right? mRNA at the 2nd carbon, it is having OH group. That is also added by the methyl group. This is called cap 1. Second nucleotide of mRNA also having a OH group. That is also added by the methyl group. This is called cap 2. Now, what's the point you all people has to remember? Is it cap 1, cap 2? All are necessary? No. Cap 1, cap 2, all are not necessary, friends. What is necessary? Cap 0 is necessary, which means what I'm trying to explain to all of you is at least one nucleotide of the mRNA should be methylated. That's the point. Why at least one nucleotide of the mRNA has to be methylated? Friends, whenever it gets methylated, no one can degrade the mRNA. Always remember, methyl group will give a lot of protection. Protection from the degradation, protection from the cleavage. That's why if the first nucleotide is added by the methyl group, enough. We can protect the entire mRNA. Now all the students try to understand. If you understand this particular slide, the capping is very, very clear to all of you. Now remember, in the process of a 5' capping, friends, there are total 3 enzymes are involved. The first enzyme, we call it as nucleotide phosphohydrolase enzyme, widely called as RNA triphosphatase. I have told to all of you many times, kinase is the enzyme which add the phosphate group. You know that very well. And you also know very well, phosphatase is the enzyme which remove the phosphate group. You know that very well. Now the question is very clear here. Listen my statements. Why did I use the word RNA triphosphatase? Because it is going to remove 3 phosphates. That's why we have given the name triphosphatase. Then the second enzyme called as guanylyl transferase. What is the meaning of the guanylyl transferase? Which transfer the guanine. Next one is called methyl transferase, which will transfer the methyl group. Guanylyl transferase, methyl transferase, and triphosphatase. Now let me explain you the process very clearly. Just pay attention and not to get confused with the concept. Look into this. Look into this. What's our ultimate goal? See, look into this, everyone. This is the mRNA, right? This is the mRNA. mRNA in the first nucleotide at the 5th carbon, at the 5th position. How many phosphates are there? Look into this. Alpha phosphate. You have to remember the name of the phosphates also compulsory. Alpha phosphate, beta phosphate, and gamma phosphate. You got that point. Now to this, what should I add? I have to add the guanine, nothing but GTP. You also know very well ZTP also having how many phosphates? Alpha phosphate, beta phosphate, gamma phosphate. But what's the point? The point is that we do not require the 6 phosphates. Out of the 6 phosphates, we have to add only 3 phosphate. Now in the exam, they can ask the question, which phosphate is getting cleaved from where? Now, what is the first enzyme? Phosphohydrolase enzyme, also called as triphosphatase enzyme. Now you see what it is doing. Look into this. From the mRNA, this triphosphatase enzyme, listen everyone, it is cleaving gamma phosphate. What phosphate? It is cleaving the gamma phosphate. So that gamma phosphate released in the form of PI. You got the point correctly. Now, how many phosphates are left in the mRNA? Tell me. Beta phosphate, alpha phosphate is present. Correct, na? Now the same triphosphatase, it is going to cut 2 phosphates from the GTP. What phosphate? Beta phosphate, gamma phosphate. The 2 phosphates can be released in the form of a PPI. Please don't get confused with that particular point. Easy to understand. No need to get confused with that particular point. Again, I am saying to all of you, try to understand. Phosphohydrolase or triphosphatase is the enzyme which cut gamma phosphate from mRNA released in the form of PI. The next, same enzyme will cut beta and gamma phosphate from GTP released in the form of a PPI. Now you tell me, guanine contain how many phosphates? You can see, right? Guanine contain only 1 phosphate. You can see that. Guanine alpha phosphate. Now that guanine alpha phosphate, every one of you remember, it will come and added to this beta alpha phosphate. This also they will ask the question, which phosphates removed and which phosphates will be added. From the mRNA, gamma phosphate is removed. From GTP, beta and gamma will be removed. In the GTP, alpha phosphate is left out. In the mRNA, beta and alpha left out. So now you see this. Then who will bring this particular guanine? So this is a beta and alpha phosphate of mRNA. This is what phosphate? Alpha phosphate of guanine. Now the question is, friends, who brought this particular guanine alpha phosphate and added it to this mRNA? That's the question. The enzyme is guanylyl transferase enzyme. Guanylyl transferase enzyme bring this particular alpha phosphate and add it to this particular mRNA. So that is capping. But we discussed, right? The guanine compulsory at the 7th position, it is added by the methyl group. That's a very important point. Now, who will add this methyl group? See the name of the enzyme. Guanine 7-methyl transferase. What was that? Guanine 7-methyl transferase is the name of the enzyme that will come and add the methyl group to the guanine. Then guanine will become so powerful. Now the question is here, see, guanine 7-methyl transferase is just an enzyme. It will just transfer the methyl group. Now the question is that who will donate the methyl group? There should be some donor. There should be some donor. Who is the donor of the methyl group? Just remember the name of the donor enough. Adomet. Adomet means adenosyl methionine. This is the one which donates this methyl group. So by taking this methyl group, guanine 7-methyl transferase enzyme add that methyl group to the guanine. That's a simple point. Upon donating the methyl group, adenosyl methionine converted into homocysteine. You no need to remember that. Don't confuse. Don't worry about it. So let me summarize to all of you very quickly. First, there is an important enzyme called triphosphatase enzyme, which will cut gamma phosphate from the mRNA released in the form of pi. Same enzyme will cut beta and gamma phosphates from the GTP released in the form of PPI. So now this guanine with a single alpha phosphate will be taken by the guanylyl transferase enzyme and add to the beta and alpha phosphates of the mRNA. Then what's going to happen now? Now, this particular guanine added by the methyl group by an enzyme called as guanine 7 methyl transferase, and methyl group is donated by Edomet. This is called 5' capping. Okay, in the next class, I will finish the tailing as well as the splicing. Okay, is it clear everyone so far? Okay, compulsory keep practicing the question. I want you people to focus on practicing the questions whichever I have given to you. Don't neglect, solving the question. Okay, because you are already having a knowledge on the subject, you put your effort in solving that particular question. If the questions are not coming from whatever you solved, whatever I explained you, questions come from there. Okay, so think and answer that. Okay. Perfect. Okay, I'll see you in the next class. Okay, please revise it once again without fail. I don't want you to read all the points. You don't have that much of time. Just keep the PowerPoint with you and keep reading point by point. Within 15 to 20 minutes, try to finish the entire PPT. Don't waste your time reading all the books. Now you don't have that much of time. And first of all, you stop Telugu Academy currently, because you already know that Telugu Academy, you already read it. So I want you to read the concepts which are not there in the Telugu Academy. Always remember and follow that. Okay. I'll see you in the next class. Thank you.